Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled Multidomain Biologics, Bioanalytical Strategies and Insights. My name is Samaya and it's my pleasure to be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, you can contact me at any time by sending a message here. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank BioAgilytix who developed the content for this presentation. BioAgilytix is a trusted partner for bioanalytical services throughout every phase of the drug development process. As a leading global bioanalytical laboratory, the company provides established and emerging pharmaceutical and biotechnology organizations with pharmacokinetic, immunogenicity, biomarker, and CMC analytical testing services in a GXP environment. BioAgilytix maintains state-of-the-art laboratories in Durham, North Carolina, Boston, Massachusetts, San Diego, California, Melbourne and Brisbane, Australia, and Hamburg, Germany. Now it's my pleasure to introduce our speakers for today's event, and our first speaker is Lynn Kamen. Lynn Kamen is a scientific officer at BioAgilytix. She earned her PhD in immunology at the University of Michigan and completed a postdoctoral fellowship in immunology at the University of California, San Francisco. Dr. Kamen has over a decade of experience working in drug development, from early target discovery through clinical development for both large and small molecules at several companies, including Portola Pharmaceuticals and Elector. More recently, she was a principal scientist at Genentech, where she supported the in vitro biological characterization of large molecules and led the development of immunogenicity assays, including ADA, NAB, and immunogenicity risk ranking assays. And our next speaker is Dr. Christopher T.J. Dr. Christopher T.J. is an associate director and principal investigator at BioAgilytix Europe, Hamburg, where he oversees the conduct of pharmacokinetic, immunogenicity, and biomarker bioanalytical assay establishment, validation, and sample analysis in support of sponsors' drug development programs. Chris obtained his diploma degree in biochemistry from the Leibniz University, Hanover, and then earned his PhD in biochemistry and molecular biology at Kiel University, so that's CAU. Our next speaker is Joshua Kabison. Josh Kabison serves as a Manager 2 of Bioanalytical Operations at BioAgilytix, where he oversees the development, qualification, and validation of bioanalytical methods in support of large molecule drugs. He has worked in the regulated CRO space for over 12 years, including the last 8 years at BioAgilytix. While processing experience across several platforms, Josh's specialty lies in cell-based immunogenicity assays, such as neutralizing antibodies, so that's NAB, and enzyme-linked immunosorbent spot assays. And then our final speaker is Liz Nolf. Liz Nolf has over 20 years of assay development and validation experience with various modalities and therapies, cell and gene therapy, enzyme replacement, monoclonal antibodies, and lipid and hormone small molecule ligand binding assays. Nolf serves as Manager 2 BPM at BioAgilytix, servicing a strong client portfolio specializing in immunogenicity, PK, oligohybridization, LBA, and biomarker studies. She previously worked for Enzo Life Sciences and Kamen Chemical, identifying emerging areas of research, diagnostics, and drug discovery, and implementing products of interest to various disciplines into those pipelines. So now, without further ado, I'd like to pass control to our speakers. So Lynn, whenever you're ready, you can go ahead and get started.
Thank you so much for the wonderful introduction, Samaya. Greatly appreciated. And I think we're just pulling up the slides now. Yep, so today we are going to be discussing multi-domain biologics, sharing both bioanalytical strategies and insights into this complex modality. Next slide. So as a high-level overview, we're going to start with understanding what are multi-domain biologics, giving an overview of their structural complexity, mechanisms of action, and the therapeutic potential beyond traditional monoclonal antibody therapeutics. We're then going to discuss the bioanalytical challenges, how multiple domains can create unique considerations for assay design, safety evaluation, and efficacy assessment. We'll discuss assay development and validation strategies, including best practices for creating pharmacokinetic and immunogenicity assays that capture the full therapeutic profile. We're going to translate insights to clinical development, sharing guidance on when to apply additional molecular characterization to support regulatory success. But first, we're going to start off with a general introduction of what is BioAgilytix. Well, we are a global CRO with the size, scale, and scientific expertise to meet all of your bioanalytical and CMC analytical needs. Next slide. Our headquarters are located in Durham, North Carolina, and we have sites with comparable capabilities in Boston and Hamburg, Germany. Our site in San Diego specializes in LC-MS and is one of the largest mass spec labs on the West Coast, whereas our Australian subsidiary specializes in bioanalytical support for first-in-human trials across all modalities and platforms. And as a dedicated bioanalytical lab, we offer the services of pharmacokinetics, or PK, immunogenicity, biomarkers, and CMC analytical. Next slide. These core 4 services are supported by a number of different platforms, ranging from ligand binding assays, cell-based assays, LC-MS, and molecular technologies. We support the entire drug development pipeline, all the way starting from early stage drug discovery through post-marketing commercialization. Next slide. So as I mentioned before, today we are going to be discussing bioanalytical strategies for multi-domain biologics with case studies detailing our experiences and lessons learned from developing both pharmacokinetic and immunogenicity assays for these complex modalities. However, before we get into the more technical aspects, I want to introduce what are multi-domain biologics for those in the audience who are new to this modality, to help everyone get a better understanding of why the bioanalytical strategy is so unique. Next slide. So, multi-domain biologics are, simply put, an engineered biotherapeutic composed of 2 or more distinct domains, each contributing a unique function to the drug's overall activity. Multi-domain biologics can take several forms, included but not limited to bispecific antibodies, trispecific antibodies, and fusion proteins. Due to the multiple targets and delivery capabilities, multi-domain therapeutics have potential for enhanced therapeutic efficacy as compared to traditional monoclonal antibody therapeutics. As such, this modality is rapidly growing, and shown here is a snapshot of the different recent approvals for multi-domain therapeutics that have received FDA approval since 2024. To better understand the rise of this new modality, let's dive into their benefits. Next slide. As compared to a monoclonal antibody therapeutic that binds to one target, exerting only one functional effect, multi-domain biologics have the potential for enhanced efficacy by engaging multiple targets. They offer targeted delivery of functional activation. Common multi-domain biologics are T-cell engagers that engage T-cells to become activated against specific target cells, such as tumor cells, shown here in this figure to the right. As a result of this targeted activity, they have the potential for reduced side effects, greater versatility than mono-targeted therapeutics, and can offer improved PK profiles. Multi-domain biologics can also be more cost-effective as well. By having one therapy exert multiple effects, you can decrease the total number of therapies needed for treatment. However, due to their complex structure, they do require specific bioanalytical strategy. Next slide, which is depicted in this slide right here. During preclinical studies, the predominant bioanalytical readout is PK, or drug exposure. How much of that multi-domain biologic is in circulation? As you move into regulated studies, like the GLP tox study, assessing the immunogenicity or anti-drug antibodies is important to check to understand the PK profile, since ADAs can lead to clearance of the drug from circulation. In today's presentation, we will discuss how the PK assay itself changes from preclinical to clinical studies, and then we will also dive into how ADA characterization, including domain characterization and neutralizing antibody testing, can lead to enhanced understanding of not only the safety, but the efficacy of your multi-domain biologic. So to get us started, Chris is going to discuss, next slide, PK strategies for multi-domain biologics. Chris, please take it away.
Thank you, Lynn. So please, next slide. So it's, yes, so pharmacokinetics. So what does it mean and what we're talking about? So pharmacokinetics usually stands for PK assays and quantitatively measuring the concentration of a drug in the body. And it's part of studying the bodily absorption, distribution, metabolism, and excretion of drugs. And it helps to understand the fate of a drug administered to an organism. It would also provide information important for establishing dose levels, exposure, dosing schedule, formulation, and a route of administration. On the right-hand side of this slide, you would see a typical simple PK profile, where you can see the Cmax, so the maximum concentration of a drug in a body at a certain time, also called Tmax. And you can also, in the elimination phase, you can determine or calculate the half-life of the drug over time. The next slide, please. So how do we tackle multi-domain biologics in the PK? So first of all, let's look at the preclinical PK. Preclinical PK assays often use commercial reagents that target the antigen, or we're using generic anti-human antibodies to capture and detect the drug product. A generic approach uses often anti-human IgGs to both bind and detect the drug product. We can also see it here in the first scheme in the middle of the slide. To achieve higher sensitivities, you can use combinations of a target antigen capture and a detection, which is then showed here on the right-hand side of the slide. Next slide, please. So what's the difference to a clinical PK? Due to high levels of IgG in circulation and the need for sensitive assays during the first-in-house studies, clinical PK assays often use custom critical reagents, such as anti-idiotypic antibodies, that bind specifically to each domain of the therapeutic. A combination approach with polyclonal antibodies and antigen capture or detection is also possible. Often, the use of commercially available reagents can be very expensive over the time of the study and for the entire clinical study. On the right-hand side of this slide, you see a schematic clinical PK assay using anti-idiotypic antibodies for capture and detection. Next slide, please. So let's dive into a case study for the development of a PK assay for a trispecific antibody. So let's call this trispecific antibody drug T, and let's have a look how it looks like. We see a heterodimeric Fc targeting 3 different receptors called target A, B, and C. The binding of these receptors induces target cell activation and subsequent therapeutic benefit. Next slide, please. So the PK assay for this type of molecule used a pair of anti-domain antibodies to capture drug on the plate. It's recommended to test different combinations in checkerboard style experiments to really determine the optimal pair for capture and detection. Reconstitution of the drug in this specific case was found to be extremely crucial. Often, alternative formulation buffers and storage conditions may be needed for multi-domain therapeutics. It's also important to think about tight control on reconstitution that can heavily improve the assay robustness. And here, as an extra, it's also a very important point to consider, coating and detection solutions were prepared always freshly. In total, multi-domain biologics can have particular preparation, buffers, and storage needs given the high degree of engineering of these therapeutics. The next slide, please. So how did that work out in the end? So overall, with the assay described for this case study, initial validation was successfully completed following the ICH M10 guideline. However, when the first in-house clinical samples were tested, it was revealed that the assay curve was not suitable and sensitive enough. So we had to redevelop the entire assay in a kind of partial validation to obtain a higher sensitivity. So the first fix to this problem was to increase the sensitivity by increasing the MRD by around 4-fold. With that, it was able to improve the sensitivity of the assay to below 1 nanogram. It can also be a point of consideration that if increasing the MRD does not result in sufficient sensitivity, one could think about utilizing or using a mix of detection antibodies against different targets, such as target A and B here, while still capturing target C. Next slide, please. So to sum up, the key considerations for PK assay establishment for multi-domain biologics is that due to the higher exposure and lack of matrix interference in preclinical animal studies, a generic capture and detection, such as using anti-human IgG antibodies, are appropriate to use for these PK assays. However, in clinical PK assays, one often requires custom critical reagents, such as anti-ID antibodies, to detect the different domains of the therapeutic. When designing the assay, it often involves checkerboard-style experiments, comparing different combinations of capture and detection, which can be quite elaborate. However, this would ultimately lead you to find the optimal pair and to find the best sensitivity, robustness, and selectivity settings for your assay. The complexity of assembling multi-domain molecules can lead to potential instabilities in solution, leading to required additional control steps when reconstituting, for example. The increased assay sensitivity for molecules with low therapeutic windows can be handled by either adjusting the MRD or trying different detection strategies, such as aiming to detect both arms in one single step. Next one, please. And this is now the part for the ADA strategies for multi-domain biologics.
Thank you so much, Chris, for sharing insights into the PK strategies. And so next, Liz is going to be talking through our experience working on ADA assay development and validation for multi-domain biologics. Liz, please take it away.
Thanks, Lynn. So for immunogenicity assessment of multi-domain therapeutics, it follows a very typical immunogenicity approach, a three-tiered strategy where the first tier is screening for ADA, targeting a 5% false positive rate, which is designed to minimize false negatives. The second tier is the confirmatory tier, which is a specificity tier designed to target 1% false positive rate. And then the third tier is characterization of the confirmed positive responses, giving a titration or a qualitative response to the molecule. Next slide, please. An ADA strategy for a bispecific antibody is shown below, in which case a developed bridging ADA is targeted against the whole molecule, the whole bispecific antibody, using a polyclonal or a positive control that was generated against the entire molecule. Next slide, please. developing the standard bridging with the whole molecule approach. We can also utilize the whole molecule in the confirmatory approach to see if the ADA response is confirmed positive towards the whole molecule, followed by adding each domain in independently, as depicted here with the red and the yellow domains, to detect which part of the molecule is the ADA response driven by. Next slide, please. In this case, if the domains are added one at a time in excess, we can determine if the confirmation of the ADA response is towards domains A or domains B, depicted by the red and the yellow again. If we see a drop in signal in one domain, it can be inferred that the ADA response seen in the core assay is directed against that particular domain. Next slide, please. So there are some pros and cons for the domain competition approach for immunogenicity assessment. There are some key requirements that individual domains are readily available, purified for each piece of the molecule. It is ideal that each domain also has a specific positive control. Domain reagents are readily available in some molecule formats, but can also be extremely challenging to produce in other formats. It may also be possible to use a polyclonal approach, in which case antibodies against each domain, as well as the core molecule, are pooled together to create a polyclonal. The pros of this approach is it's relatively straightforward. Most of these immunogenicity assessments are already designed to detect the whole molecule. And in this case, you would be just modifying the conformation tier or adding onto it to detect those domain-specific pieces. The cons, this approach can lack sensitivity to detect low levels of domain-specific antibodies, especially if there's a high prevalence of ADAs to other domains in the samples. Next slide, please. I'm going to present a case study on a multi-domain therapeutic. We're going to name this drug A. Drug A is comprised of 3 domains, domains A, B, and C. And we have available recombinant proteins of each domains, A, B, and C. We have a polyclonal directed towards the entire drug A molecule, as well as positive controls targeted to domains A, B, and C independently. Next slide, please. The standard immunogenicity assay setup is employed here, in which case we have a bioatinylated and rethinylated drug A used as the capture and detection or the bridging mechanism, and the polyclonal to the entire molecule drug A is used as the surrogate positive control. In this case, it's a bridged format in which the bioatinylated drug A binds to the SPC, and the rethinylated drug A also binds to that SPC, forming a bridge. Next slide, please. In the initial domain characterization, we want to test if the drug A whole polyclonal SPC will bind to each domain independently. In this case, we will add domains A, B, and C to see if we can compete off the domains or the whole molecule utilizing the domain of interest. So in this case, the first figure, domain A was added into the confirmatory tier using, again, the whole molecule SPC. And we can see that it does, in fact, bind the SPC and prohibit binding of the rethinylated drug A. So we see inhibited response. Domain B, much the same. We see that it does bind the whole molecule SPC, inhibiting the detection reagent. However, in domain C, we do not see this inhibition. So it can be inferred that the whole molecule SPC does not bind well or directly to domain C. Next slide, please. This is just inferring that. And then, so to come around this, due to the complex structure of drug A, it's not known if all domain-specific SPCs can bridge in the parental ADA assay. meaning the polyclonal towards the whole molecule can form the bridge, but can polyclonals or SPCs generated targeted towards each individual domain bridge the whole molecule? To test this, we added in the domain-specific positive control to the whole molecule parental assay. And you can see here with domain A, it does bridge the whole molecule just utilizing the domain A positive control. Also, we see inhibition of signal adding in domain A into the assay. Next slide, please. Domain B, much the same, added into the parental assay. We do, in fact, get bridging of the detection and capture reagent whole molecule, as well as inhibition of the signal in the confirmatory formats. Next slide. Domain C, in which case we did not see the inhibition or bridging using the whole molecule positive control. With the domain C specific positive control, we do also see bridging of the parental assay, and we do also see inhibition of the parental assay utilizing the drug C specific positive control and adding in domain C in the confirmatory format. Next slide, please. There are some key takeaways for domain characterization of multi-domain biologics. To validate the domain characterization of drug A and use domains A, B in the original parental assay, we would add in domains A and B into the confirmatory tier, and if neither domain confirmed positive, it could then be inferred that the ADA was targeted to domain C. Alternatively, we could have a confirmatory assay where domain C specific positive control is employed, and if we see inhibition in this format, we know that the ADA response is driven to domain C. Domain characterization is a valuable tool for understanding immunogenicity to multi-domain therapeutics. It can help us understand if there's one component of the molecule that's driving immunogenicity response more than others. It can also help lead to improvement of future multi-domain biologics in the pipeline, especially if they're using the same framework or the same backbone of a molecule. To properly characterize ADA against multi-domain, really the key is critical reagents and the availability of those reagents. Recombinant derivatives of each domain is required. SPCs against each domain is ideal. And the more critical reagents that we have available, the more robust the immunogenicity testing strategy can be. Next slide.
Thank you so much, Liz, for taking us through your experience working on ADA assays for multi-domain biologics. And now to finish out the trifecta of immunogenicity analysis, we have Josh here to describe strategies for detecting neutralizing antibodies to multi-domain biologics. Josh, take it away.
Perfect. Thanks, Lynn. So I have the distinct pleasure of presenting the final characterization tier of the immunogenicity response, which is generally neutralizing antibody assays. So these are our strategies for detecting those NAbs against multi-domain biologics. Next slide, please. Yep. So in general, there's multiple formats for NAb assays. We tend to break those into two distinct buckets: those cell-based or competitive ligand binding NAb assays, which are plate-based NAb assays. For the most part, what we're seeing is multi-domain biologics fall into the cell-based functional assay readout. Next slide, please. There's multiple different common cell-based NAb assay formats. We have proliferation assays, which involve the addition of drug to a cell-based plate, causing proliferation of those cells, neutralizing antibodies to that drug, then reduce that ability of the drug to cause proliferation, thereby NAbs are detectable via a decrease in luminescent assay signal. We also have apoptosis assays, which are very typical for maybe an ADC molecule, where addition of the drug to a tumor cell line causes apoptosis of that cell line. Neutralizing antibodies would then be blocking that drug activity, failing to cause the apoptosis. So your typical cell titer glow readout, for example, would be detectable via an increase in luminescent signal. Next slide, please. For our typical assay for a bispecific antibody, and in this case study, we have drug P here. We have our standard T-cell engaging molecule. So most of these molecules target a tumor cell line for a cancer therapeutic and engage a T-cell that generally has a luciferase reporter gene present within the T cell. The bridge formation by drug P between the tumor cell and the T cell causes activation of the luciferase reporter gene within the T cell line. That causes an increase in luminescent signal detectable via the drug activity. Neutralizing antibodies to either side of drug P's both bispecific domains causes blockage of the bridge formation between the tumor cell line and the T cell line. Blockage to either side of drug P and the lack of bridge formation does not allow for activation of that reporter gene, thus causing a decrease in luminescent signal if NAbs are present in the well. Next slide, please. So with bispecific molecules, it is important to have an SPC, a surrogate positive control, that is specific in terms of the antibody therapeutic. It's just similar to the ADA. That allows for dual detection of both sides and proof of concept that you are able to detect those NAbs to both sides of the bispecific antibody. In this case study, we actually were able to demonstrate both of the neutralizing capabilities to both arms of the bispecific molecule, but our CD3 arm, which is the T cell arm of the molecule, the SPC did not have adequate sensitivity to hit our general target to that arm. Our sponsor was able to provide a polyclonal SPC to the entire drug that had very good sensitivity and allowed for use of that polyclonal as the assay surrogate positive control. An alternative strategy to this would be, though, to create an antibody cocktail using both arms of the SPC, or both arm SPCs, mixing those at equivalent molar ratios, and then using that as a cocktail SPC for the final assay validation. We've also seen full validations using both arms completely independently as well. One additional issue we had in this assay was high levels of circulating drug target within patient samples. The disease state had circulating target that got in the close to 1 microgram per mil level within patient samples, thus causing an extra issue as circulating target could actually cause false positives in the assay. Next slide, please. So what is common for multi-domain bispecific antibodies is the need for a bead-based method for pretreatment. Because these bispecific molecules are generally antibodies, they have a long half-life, which causes high circulating level of drug within the samples. And on top of this, we also had the soluble target that we just discussed, which also created a second interference problem within the assay. Anyone that's worked on cell-based assays for NAbs generally knows that drug interference can become a very big issue. So our first step was to introduce a purification method to remove the excess drug within patient samples. This was done by using a streptavidin-coated bead and capturing a biotinylated drug onto those beads, acidifying patient samples to dissociate the ADA and the drug within samples, using that biotinylated drug, streptavidin-coated beads, and adding those back into the system after acidification with our neutralization buffer, and allowing for outcompeting of the free drug with the biotinylated drug. In addition, we implemented a high concentration of anti-target antibody within the system to saturate the soluble target and wash it away as we perform our streptavidin-coated bead washings, which have now captured the free ADA within the system. That saturating level of anti-target antibody essentially removed all presence of soluble target within the system, thus reducing that risk of false positives. So kind of killed two birds with one stone. We were able to remove the drug interference from the assay, as well as remove the target interference from the assay, thus having a much cleaner, much more sensitive readout with freedom from false negatives and false positives. That final step after the washing is simply acidify the ADA that have now been bound to the biotinylated drug back off of those streptavidin-coated beads, and thus we have our free ADA in the supernatant, which can be neutralized and analyzed in the NAB assay. Next slide, please. Yeah, so key takeaways for neutralizing antibodies for multi-domain biologics. Given their MOA, the multi-domain biologics are often cell-based assays. Again, these are molecules that are engaging a T cell and a target cell line, typically a cancer cell line, at the same time. Thus, two-cell MOA makes a great use of a cell-based assay where you can replicate that very well in vitro. You can use polyclonal antibodies or a combination of anti-idiotypic antibodies as your SPC. Ultimately, you want to make sure you're demonstrating the highest degree of sensitivity possible. It is critical, as we observed in our assay for this molecule, that you understand the biology of your patients and the biology of the therapeutic, so you know the potential sources of interference. And that is critical for, I would say, all molecules, but definitely in this case as well. And as with any cell-based NAB assay, sample pretreatment methods very often have to be implemented in order to circumvent matrix interference, but as well as high levels of circulating drug in your patient samples. They often are timely and costly to implement, but with them you can achieve a much better output of your NAB data. Next slide, please.
Thank you so much, Josh. And so I think just to conclude this part of the webinar, hopefully you have gained an understanding that due to their multimodal nature, multi-domain biologics present unique bioanalytical challenges. PK assays for these molecules oftentimes require custom critical reagents, either target antigen or anti-idiotypic antibodies, to determine the pharmacologically active form of the multi-domain biologic. Immunogenicity analysis can involve additional characterization tiers because of the complex structure of these multi-domain biologics. So in addition to running confirmatory assays with the entire drug, using separate drug domains can be helpful to determine if the ADA is binding to one region or another of the drug product. And it is important to fully characterize all the critical reagents in your immunogenicity analysis to better understand the potential outcomes. Understanding the biology, i.e., whether or not circulating target is present, is critical to making a reliable immunogenicity assay. And with that, I think we can go to the next slide. Open the floor for any questions that you might have. Thank you, everyone, for your attention.
Well, thank you very much for that insightful presentation. And yes, that does bring us to a Q&A portion of this webinar. So attendees, if you have questions, keep sending them in. We've already received some questions, so let's start off with those. And our first question here is, for the PK, why did you exclude moiety B? Isn't it important for the quantification of the intact molecule?
Chris, I don't know if you want to take that question.
Would you please start?
Of course. The question was, for your PK case study that you described, why did you exclude the B region of that molecule in your capture and detection? Isn't it important to quantify whether or not the entire molecule is present?
Yeah, I mean, with the specific capturing of the, let's say with portion A, you should have B and C, right? And then it's just a matter of detecting. So that was our assumption. So we tested different combination and found out that this is the absolute best and most sensitive assay we could obtain with this combination. So it had nothing to do with like omitting B. It was just a matter of testing.
Understood. Thank you so much.
So
Great.
yeah, just to give some side information, there was quite some effort, right, to test all different combinations. So it is also for ADA and NAB. So that's quite a lot of effort. But in the end, if you want to reach the best sensitivity, the best robust assay, so it's worth the effort.
Great, thank you, Chris. And we have another great question from an audience member. The question is, for preclinical PK assays, is it better to use custom critical reagents?
Yes, so maybe I just also take that. Yes, in my opinion it is. So just imagine, I mean, preclinical assays ideally help you to pave the way for the clinical assays. And if you already have like good reagents that can be used in a clinical assays at hand, this makes the, let's say, transfer from preclinics to clinics way more easy and effective. So that is why we usually recommend in having the best reagents possible at early stages. It's not always possible, that's for sure. Things take a while to create these custom reagents, but if possible, that would be our preferred way, and this would be our recommendation, as said, because the transition from preclinics to clinics is way smoother then.
Wonderful, thank you again, Chris. Another great question here is, for multi-domain molecules with masked domains that cleave in vivo, how would you approach the ADA and NAB analysis?
I can start that one off. So that is a challenge for multi-domain therapeutics as far as where they're designed to be either cleaved in vivo or where they are linked together to form the complex. Those domains can be masked to what a antibody would be able to see or an immune response would be able to generate. Ideally, in a perfect world, we could have the critical reagents characterized and synthesized to contain both the masked and the unmasked, so what it would look like once the body cleaves, and be able to test both of those in the assay formats I referred to before in the confirmatory assay for masked and unmasked. So for example, we'd have a masked and unmasked domain A, B, and whatever domains are linked together and or cleaved in vivo.
Wonderful, thank you, Liz. Another interesting question here is, when do you...
Before we move on, I think one more thing that would be interesting when we're thinking about masked versus unmasked, not only for ADA, but for NAB assays, do you need to develop a NAB assay to the masked version as well as the unmasked? And Josh, I know you have a lot of experience with this.
Yeah, absolutely. So generally we think of the unmasked version as the version that is most susceptible to NAB activity and NAB detection, because generally we think of those variable regions as needing to be present in order to really neutralize the drug activity. But there are instances where we are able to actually unmask the molecule as part of the assay. and it can become a balance of robustness versus MOA. But if you are able to unmask the masked version as part of the assay, that actually can allow you to potentially detect any immunogenicity that's preventing that action from happening in vivo. But generally, we'll use the unmasked version primarily, so both variable regions of a multi-domain bispecific are available and capable of being detected for NAbs.
Thanks, Josh.
Thank you again. And another question here is, when dealing with circulating target interference, you mentioned using an anti-target antibody to clear out the target. Couldn't this also remove the NAb?
Yeah, so I'll take that one as well. Yes, that is a legitimate concern. So when you introduce an so the target that we're talking about here is the drug target, right? So when we're talking about introducing another antibody that also binds to that same target, there is concerns that the CDRs from the drug as well as the anti-target antibody could have some overlapping regions, some similarities that actually will allow our NAbs in samples to bind to that anti-target antibody. So it's not a very, very high risk issue, but it's something that does need to be checked against. Simply checking the sensitivity of the assay with and without the presence of that anti-target antibody is a way to quickly and easily see if that is an issue or not an issue. We were able to do that, did not see any interference from our anti-target antibody, but that is a critical step to make sure your anti-target antibody is non-competing for your NAb activity.
Fantastic. Thank you again, Josh. And another interesting question here is it required to perform domain characterization for multi-domain biologics? If not, when do you recommend starting the extra characterization?
You know, that is a really great question, something that we get asked a lot of time. And it's kind of an open question. There is no right or wrong answer. So if you look at the 2019 FDA immunogenicity guidance, it says that domain characterization for multi-domain biologics is recommended, but not necessarily required. So this is really a very project or sponsor specific question to answer. So I would recommend having it in all of your pivotal or registrational studies, because it really does help to flesh out your entire immunogenicity package that will be part of the integrated summary of immunogenicity during your final filing. But the question is, during early stages like your phase 1 or phase 2, how important is it to bring on this domain characterization? I think of it as in a couple different ways. One, if you have a pipeline of multi-domain biologics that are all on the same framework or all use the same arm, it might be helpful during your early clinical studies to do domain characterization then, because you're going to quickly find out if there is a problem with one particular domain or another, which might help you make changes to the molecules that are coming up later on in your drug development pipeline. But if it is only a one-off multi-domain biologic that you're working with, you might elect till later studies, such as phase 2 or phase 3, to start to build in the domain characterization. And another layer to that question as well is, does the domain characterization need to be fully validated in a GCLP fashion? And that really is another open-ended question. What we find is during early stages, it can be run as more of just a characterization, kind of a research use assay, but then during later studies is when you might want to fully validate that aspect of it. And Liz, I know you've experienced doing work, working with both the characterization end of domain or the more research end of domain characterization, and the more regulated end of domain characterization. Yeah,
definitely. You know, we've seen it at all levels of the drug going through a trial. Just to add on to that, I think the critical reagents, again, are the most key pieces to developing these assays, and oftentimes those can take quite a while to produce. So perhaps kind of even starting to think about it in your initial phases, even if we're not going to employ it until later phase trials, it can help us to build and model the assay that will be able to do those domain-specific characterizations, even if we're going to hold till some more of the pivotal trials.
Mm-hmm. Agreed. And Josh, domain characterization in NABs? Yes, no, maybe so?
Yeah, generally you want to at least show that your NAB assay is capable of detecting NABs to both sides. Now, not every sponsor has the luxury of generating a PC to both sides. Some sides, particularly the T-cell side of most multi-domain bispecifics, is generally challenging to get a good SPC against. So there are surrogate ways like dropping a cell line to show that your assay can detect that has signal changes if you block out that side of the assay. But generally, in a perfect world, we at minimum have SPCs to both sides to show that the assay is capable of detecting NABs to both sides. But it's not something we can't overcome if we only have one or the other.
Good to know. Thank you.
Wonderful. Our next question here is, can you describe in more detail what you mean by the dual antibody approach for increasing sensitivity of PK assays?
Yeah, I'm also happy to take that one. So, so what we mean with that is that you actually use a mixture of two antibodies for detection. So in this example of a trispecific, you, let's say, capture with targeting domain A, and you would then detect B and C in a mix of two different antibodies that are both labeled. So that's something that can heavily increase the sensitivity of the assay, mimicking kind of polyclonal response, right? So you have like different antibodies in an ADA, for example. So you would just increase sensitivity by that by mixing these two antibodies. And then just maybe also adding to the previous point, so what I learned and what I recommend, so it of course depends on where you are and what kind of reagents you have at hand, but it's also a safety consideration, right? So what does your trispecific target in a cell, in the body? And is there somehow endogenous counterparts? In some instances, we have seen that you know, we have like an endogenous counterpart that could be also targeted by any potential ADA or NAB, which is then of course a very risky thing. And if you observe something, it would be very critical to the project. So in these instances, it makes very much sense to start as soon as possible with at least an assay having it at hand. Whether it's qualified or fully validated, that's another question, but this is definitely also one important consideration to that topic.
Wonderful. Well, thank you everyone for sending all of those questions and for the team for all of those answers. We reached the end of the Q&A portion of this webinar, but not to worry, if you have more questions, go ahead and quickly jot down everyone's emails here. So go ahead and do that now. And if you do obviously have further questions, you can direct them to the email address, or if you just didn't get a chance to get to your questions, the team at BioAgilytix will follow up with you later on as well. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues, so I encourage you to do that. Now, please join us in giving a big thank you to our speakers today. So that's Lynn Kamen, Christopher TJ, Josh Kabisen, and Liz Knopf for that fantastic presentation. We hope you found this webinar informative. Have a great day, everyone.
Thank you.
Thanks all.